All right, let's jump right in. Imagine, if you will, a high-tech manufacturing factory. But we're not talking about building cars or smartphones here. No, we're talking about building life itself, and the stakes, well, they could not be higher. Every single day, your body relies on literally millions of these microscopic factories operating flawlessly. So in this explainer, we're going to completely decode the intricate, heavily guarded production line that we know as the cell cycle.
It really all boils down to this one absolutely thrilling question. How does a single cell become two perfect copies without making a catastrophic mistake? Seriously, think about it. When a cell divides, it doesn't just, neatly split in half. It has to precisely duplicate its entire genetic manual, sort it out, and package it up. And even one tiny microscopic error in this factory can lead to disastrous consequences, like cancer. The tension is incredibly high. So, how exactly does the cell pull this off?
Okay, let's dive into this. To really grasp the master plan, we need to define our terms. The cell cycle is this continuous, highly coordinated sequence of events, starting from the end of one nuclear division straight through to the beginning of the next. And the overarching strategy here? Absolute fidelity. The cell absolutely must accurately duplicate its contents, especially its genetic material, and divide to produce genetically reliable daughter cells. It is, without a doubt, the ultimate heavily regulated production line. To make all this happen, the factory is actually running 3 interconnected cycles simultaneously. It's like a giant puzzle. First up, there's the chromosome cycle, where DNA synthesis alternates with nuclear division, also known as mitosis. Then we have the cytoplasmic cycle, which is the physical growth of the cell alternating with the actual splitting of the cytoplasm. And finally, the centrosome cycle, which precisely duplicates the structures needed to form the 2 poles that are going to eventually pull those chromosomes apart. Everything must work in perfect harmony. If one conveyor belt speeds up while another slows down, the whole system crashes. Looking at the big picture of our factory floor, the cycle is broadly divided into 2 main parts. We've got inner phase, where the cell is busy growing and copying its DNA, and the mitotic phase, where the actual division happens. And honestly, the most striking part, the actual splitting, that mitotic phase, is just a tiny fraction of the total cycle. The real work, the vast majority of the time, is completely dedicated to the preparation. Let's break down that preparation period a bit more. Interphase eats up about 95% of the cell's time, and it happens in 3 distinct shifts. First is the G1 phase. Here, the cell is metabolically super active, growing, synthesizing RNA and proteins. It's gathering resources and deciding at this crucial restriction point, or start point, if conditions are actually right to commit to dividing. If not, it might exit into a dormant resting state called G0. Next is the S phase, or synthesis phase. This is where the magic happens. The cell replicates its DNA, doubling its genetic content, so each chromosome now consists of 2 identical sister chromatids. Finally, we move into the G2 phase, where the cell continues growing and synthesizing the specific proteins it needs for the upcoming division.
Now, what's really interesting about the G2 phase is what's going on under the hood. It's not just about getting bigger. It's a phase of profound cellular introspection. Before the cell commits to the dramatic, irreversible act of splitting, it takes a moment to double-check its own work. It asks itself, is my volume okay? Is the environment good? And most importantly, are there any mistakes in the duplicated DNA? It acts as a pure quality control step to ensure absolutely flawless replication right before the point of no return.
Which brings us to the factory's security gates, officially known as checkpoints. The cell cycle is heavily regulated to prevent disastrous genetic errors, meaning the factory only proceeds when conditions are absolutely perfect. We've got the G1 checkpoint, checking for DNA damage, cell size, and nutrient availability before it even commits to replication. Then, the G2/M checkpoint steps in to verify that the DNA was completely and perfectly copied. And finally, the M checkpoint, or the spindle checkpoint, ensures all those sister chromatids are correctly attached to the spindle fibers before they get pulled apart.
Running these security gates requires 2 very specific types of staff. On one side, we have the positive regulators. Think of them as the managers. Their entire job is to drive the cycle forward. And the main players here are cyclins and cyclin-dependent kinases, or CDKs. On the flip side, we have the negative regulators. These are the bouncers, the emergency brakes. Their job is to halt the cycle immediately if any errors are found. The most famous of these are the P53 and RB proteins. For the factory to run efficiently, the managers need to be active and the bouncers need to step back. Unless, of course, there's a genuine emergency.
Let's zoom in on those managers for a second. CDKs are actually totally inactive on their own. They're just kind of waiting around the factory floor.
They only kick into gear when they bind to specific proteins called cyclins, which naturally fluctuate in concentration as the cycle progresses. Take the maturation promoting factor, or MPF, for example. It's a complex of a CDK called CDC2 and cyclin B that specifically triggers the cell to enter division by phosphorylating the nuclear membrane. It's this precise pairing, almost like inserting a physical key into a machine, that gives the ultimate green light for the cell to move through the security gates.
So the absolutely crucial point here is what happens when things inevitably go wrong. Enter P53. You might know it as the guardian of the genome. If DNA damage occurs, P53 springs right into action, acting as the ultimate quality control inspector. It halts the cell cycle right at the checkpoint, giving the cell a brief window to deploy DNA repair enzymes. But if the damage is too severe, if it just can't be fixed, P53 makes the ultimate sacrifice call. It initiates apoptosis, or programmed cell death. It literally destroys the cell entirely, rather than letting mutated, potentially cancerous DNA slip through the cracks. All right, so now that we know just how tightly guarded this factory is, what if I told you that scientists have actually figured out how to hack it? No way, right? But it's true. We can intentionally pause the factory floor from the outside to take a structural snapshot of the chromosomes. This external audit is a diagnostic procedure known as karyotyping. By cleverly manipulating the cell cycle, we can arrange and photograph an individual's complete set of chromosomes to check for large-scale genetic anomalies. To pull off this hack, scientists follow a highly specific set of steps, which you can easily remember with the mnemonic BPBHI. First, we start with blood collection, making sure to use an anticoagulant called heparin. Next comes planting. We culture the cells using a medium and introduce phytohemagglutinin, which acts like a molecular gas pedal, forcing those cells to rapidly divide by mitosis. Then we hit incubation, a strict hold at exactly 37 degrees Celsius for 3 days. Once they're rapidly dividing, we reach harvesting, where we intentionally arrest the cycle. And finally, staining. We typically use a Giemsa stain here to create these distinct dark and light bands on the chromosomes so they can be vividly analyzed under a microscope. Let's move to and see how this builds on that harvesting step, because the real magic there relies on a chemical called colchicine. Colchicine acts as the ultimate external pause button. It arrests the cell cycle specifically at metaphase, right at that M checkpoint. And why there? Because this is the exact moment when the chromosomes are perfectly aligned and at their absolute most visible. Once paused, scientists treat the cells with a hypotonic solution. This causes the cells to swell up, spreading the chromatids far apart so each individual chromosome can be clearly photographed and studied in detail. Now, hacking the cell cycle like this isn't just some neat party trick. It has massive, real-world medical payoffs. In clinical diagnosis, karyotyping helps identify congenital malformations super early on. For gene mapping, it allows us to localize specific genes on chromosomes. In cancer tracking, it's a literal lifesaver. For example, detecting the Philadelphia chromosome in patients with chronic myeloid leukemia helps doctors accurately predict their survival outlook. And for prenatal checks, it can analyze amniotic fluid to catch fetal chromosomal abnormalities, which helps couples understand the underlying causes behind repeated miscarriages. It is just truly incredible how understanding these fundamental, microscopic rules of cellular growth allows us to diagnose and predict our own health on a macro scale. We've mapped the factory floor, we've met the managers and the bouncers, and we've even learned exactly how to pause the assembly line. But, it leaves us with a pretty fascinating thought to end on. If we have already figured out how to pause the cell cycle to read our genetic fortunes, what other microscopic security systems are inside of us, just waiting to be discovered and hacked? Thanks for joining me on this explainer, and keep questioning the microscopic world around you.